Hello and welcome to Breakfast in Biology. This is day 39.
Good morning.
So, Mrs. Smith, I don't know about you, but I looked at the calendar today.
Mm-hmm.
We have 15 of these left. Just
15.
FYI.
That's it.
There's 3 weeks. On that last Friday, that's the last day where any new teaching, any new grades go. And then there's a couple days until the end of the school year for people to catch up on stuff. But really, 3 weeks of new instruction. 3 weeks. 3 weeks, 15 days. Those of you that are like, I'm going to get it done eventually. It's eventually. Like, it's time.
And in saying that, there's a few new names that are popping up in the comments, people we haven't seen in a while, etc. I mean, this is a good day to join because we're starting a new unit. So now you know what I mean? You're going into week 9. We're starting new material. So you should be right with us if you're in here today.
Yeah, stay with us every day. Do the couple of little things that we might ask you to do. And actually, how much work? How long do you think it will take them to do the work that we have for them when this ends today?
Oh man, hours and hours.
0. This is it today. If you hang out with us and listen today, you'll be in great shape. We're starting new stuff.
Now, if you didn't take the quiz, you got to go and take that from last minute.
Nothing that we haven't assigned that you shouldn't have done. Plenty that you haven't done that you should have. For some of you.
Hashtag truth. All
right. Who's here?
All right. Well.
Michael Jackson says I'm bad.
Dun, dun.
Michael Jackson beat it. Just beat it.
UPYT. Sorry.
It's a thriller every time he shows up.
Oh, man. All right. So, and now that Michael Jackson's in the house, how about we have Carissa, we have Darius, Gladys, Jessica, Emily, Michaela Moya's in the house, Reagan, who we have not seen for a while, Maya, Eli, Annie, Kaylee Herman is a new name to me, Abby Bossinger, Cohen, Lexi, Abby. I think I said Eli. If I didn't, I don't know if I said Darius was first, but I am flopping down the bottom. Radika's here. Our lurker Emma is here. I know there's other lurkers out there. Kirsten lurks. Is Kirsten here?
Finley Miller told me that she lurks sometimes. Finley, are you out there? Because this would be a good day for you to say hello. Stop acting like a smooth criminal.
Oh my.
And let us know you're out there, everybody. Give us a little hello in the chat. You know the way you make me feel when you do that. How many have we got so far? It's been at least 6 or 7.
I don't know. I mean, they're so easy. It's like ABC 1, 2, 3. Like
I know. I might have to phone a friend, call like Billie Jean or something to help me out. I feel like I'm talking to the man in the mirror today.
All right. Well, I guess our lurkers aren't here.
I know.
It's sad.
I know. I would like to see them.
I have all of these Michael Jackson songs now, like going in circles in my head. Like, it's terrible.
Just think when you sing those to yourself and they go through your head and we do things. It really is a good way to heal the world right now. All right, let's get down to it. Let's start talking bio. Did we not hear from? We didn't hear from Finley.
No. No.
I hope she sees this on the rerun and is sad. All right. I should share our little thing. What do you think?
It's good.
Let me just move something. All right. We've done this enough times. You think I could do it by now, but all right, there we go. Boom. Hi, Amoeba Sisters.
Woo! You know, we got to start out with Amoeba Sisters if I'm going to talk. And I retweeted this one yesterday for Mother's Day because it gave me a little chuckle. And I figured this was perfect as we're transitioning from, hey, Finley's here. From meiosis.
Can we pause for 1 second?
Yeah.
We have to, I guess, pause for 1 second because Finley said hi.
She did. So that means
happy birthday to you. Happy
Happy birthday to you. Happy birthday, happy birthday, happy birthday to you.
Cha cha cha. Cha cha cha. Happy birthday, Finley. There you go. You're back.
All right. So back to meiosis. So we finished up mitosis and meiosis. And so, you know, we kind of are moving into this idea away from meiosis, but not fully, because the next thing we have to talk about goes hand in hand with that. So I want you to start thinking, and I want you to look at the next picture and tell me in the chat if any of you recognize this man. I can't click, Mr. Suter. I'm like stalling, waiting for you to like go. And anybody recognize this man? It is not America's Most Wanted.
The cross gives it away. Some kind of religious fellow.
He has something to do with science. Man, I've seen this picture so often that like I just know it. Maybe if we go to the next one, that'll put a clue.
It would really appease me if they would know.
Okay, so Abby does, but she forgets his name. Anything that you can remember about him then, Abby, that will give us a hint that you know who it is?
The lag made that funny. 17th century Dwight Schrute. It's not. I don't know. You want the next picture?
Yeah, maybe this will, you know what I mean? You know, let there be peas on earth. Like, does that give you a hint now?
How to heal the world.
Man, either the lag's awful or we're just not punny today.
Well, I'll go back here because maybe they were just struggling because it's a black or white picture, which is another Michael Jackson song.
All right, so this guy has to do with peas.
It has to do with peas.
And he is of a religious nature. We'll go with monk.
Oh, a monk.
His name, the monk dude. Gregor Mendel Cohen for the win. And Aiden, the dude that did the genetic thing with peas.
Yes. That's
yes.
the dude.
Gregor Mendel. Gregor Mendel. Very good. So if we're talking about Gregor Mendel, what is our new unit?
Good math.
Oh, Donald Trump Big Boy is back again. It's been a while.
So if he did the square thing and the dude with the peas, yes, Emily, Eli, very good. Genetics.
Wonderful. We're going into genetics. Slide, Mr. Suter.
I'm going to get fired.
So if we think about everything that we've talked about, because we've talked about DNA, and we've talked about mitosis, and we've talked about meiosis, and now we're moving into genetics. They are all connected. They are, you know, and everything in biology is kind of connected. But these three units in particular are really things that go together. So if we think about this idea of all the way back with DNA with replication, you know, we replicate DNA in order to go through mitosis, which is to repair, for growth, et cetera, et cetera. We go through replication and then meiosis if we're trying to go and create new life. And so after meiosis, okay, then we move into having things. What are these things? At the end of meiosis, what do we get? Or rather, like, you know, what happens? Maybe we should go there first. What happens with that information, once it's replicated, what happens in meiosis?
Yeah. What are we making? Oh, it's those words. Are you looking for those words high school kids don't like to say out loud?
Well, I was starting to go there and then thought like maybe they wouldn't get there yet. So I thought they'd get something. So we copy it and we start to do what?
It creates similar cells.
Okay, well, that's mitosis. Mitosis, we want genetically identical cells.
There, Eli, got
Meiosis, we want to get our reproductive cells or our gametes or our sex or our sperm and egg cell. And so if we go to the next slide, remember that we hit that phase of anaphase, and this is, anybody tell me in the comments whether this is anaphase 1 or anaphase 2? Okay, and so we start to separate the homologous chromosomes and the chromatids from a chromosome pair. And as we do that, one of the most important things is getting genetically different cells. If you take a peek at those chromosomes there, how they have the tips, remember that's from crossing over.
Mm-hmm.
Okay, and so, everybody's saying anaphase 2, but I want you to take a look at these. What are they in? How many chromosomes are attached to one another? How many chromatids are attached to one another? So if there's 2 there,
yeah,
and I have no idea who Tukakiran is, but they're here, and they say 1.
They're right.
And they're right. Okay, so we're just in the very beginning. The second stage, we're going to separate the sister chromatids from one another to get 4. So we're going to go to individual chromatids so that we are left with 4 genetically different cells. I mean, think about how traumatizing this is for our poor chromatids. I mean, they're separated and, I mean, they really need therapy after this. Like, they're separated into individual cells. We had a lot of fun putting this together.
Well, and then, so the right answer to your first question, right?
Right, is our creating of our gametes, our sperm and our egg. And the reason that we get down to half the genetic information, if you remember from last week, is because then when they're joined together, we get back up to the original amount of genetic information. So if we have, our 23 pairs of chromosomes or 46 chromosomes, you don't want to have an offspring that has 92. We want to have the same amount. So we got to cut it in half to 23. So that when we come back together with the sperm and egg to make the zygote, we have 46.
Okay. Yeah. All right. Is this me?
This is you.
So I want you to think about, and the reason we went through this meiosis stuff a little bit is to kind of get you back into the same place. Thinking about what we're looking at, where these 2 chromosomes came from. So this picture is after this picture. So the sperm carrying half the chromosomes meets the egg carrying half the chromosomes. They come together. So what I'm looking at here are 2 chromosomes. These are going to be our homologous pairs. So once this cell comes together, it's going to go through interphase. It's going to go through prophase and all that. When we get ready to make a new reproductive cell, we would put 2 homologous pairs together. These are the homologous pairs, right? Before we have sister chromatids, if that makes sense. But what we really want you to see and look at on this is where this little band is. Because let's say blue came from dad and I don't know, it looks like red, but pink or whatever came from mom. Since they're homologous chromosomes, they carry the same genes. So they carry the trait for the color of the flower. But the top one has what it calls an allele for purple flowers, and the bottom one has an allele for white flowers. So in other words, you can have the same gene, but what it looks like could be different. And we're going to talk about that. And so we call these alleles, different alleles for the same gene. And a really good example and a really bad example. So this is a great example and a bad example at the same time. Let me tell you why. It's a great example because in humans, there's different alleles for eye color. We have brown, we have blue, we have emerald, green, emerald? We have green, we have gray, whatever. Hazel, there's actually probably more. Blonde hair, red hair, brown hair, black hair. So we have these different alleles for the same thing, for eye color or hair color. The reason this is a really bad example, and just hear it, register it, and then forget it, is because these are not Mendelian genetics. So Gregor Mendel got really, really, really lucky. He's like the luckiest dude on earth, right? Right now, we are talking about this monk that studied peas a really long time ago in biology class in 2020. And we are because he picked the pea plant. Because the pea plant has really simple Mendelian genetics. It has 2 different traits, basically. Eye color has multiple traits or multiple genes that control your eye color, right? So there's multiple genes that control your eye color. So it's not as simple as what Mendel did. If Mendel had started with humans and eye color, he never would have figured it out. He figured it out because he started with something really easy. So luckiest guy around, right? So alternative genes, alternative alleles. Alleles are different versions of the same gene, basically. And I'm trying to put this in terms that you can understand it really easy. All right. You doing this?
Sure, I can bob on. So when we talk about these alleles and these genes, and we're talking about genetics, let me just take a second to remind you, if we go back to DNA, okay, that we have DNA that is transcribed into mRNA that then goes and makes a protein. When we take a portion of DNA, that is a gene that codes for something. So that's a gene, a portion of DNA. And so as we're talking about all of the things, and we're talking about that certain like band, that's just talking about a certain portion of condensed DNA that's condensed into a chromosome. And it's in the same place for every single person on that gene. So let's say we took chromosome number 2, okay, and we told you that that was the one Mr. Suter showed, and there's where the allele or the gene for eye color. If we took mine and yours and Mr. Suter's, etc., it would be in the same place on every single human. Okay, so the reason then that you might have a different eye color than your parents, if mom gave you one and dad gave you one, how do you wind up with your own traits, has to deal with something called dominant and recessive alleles. So you have one from mom and one from dad. Those alleles can either be dominant or they can be recessive. So if you think about dominant, if somebody is dominant, they're strong, they're going to have a presence, okay? They are dominant. And so a dominant allele is going to kind of overcome the other alleles. So dominant, in the definition here, says that it's going to be expressed, meaning you're going to see that allele even if it's paired with a different allele. So the dominant is going to be prominent. A recessive allele is, it's not that it's a weaker allele, okay? That doesn't mean in terms of like evolution that recessives are always bad. It just means that you need to have 2 of them in order for the recessive allele to show. So when you go to eye color, okay, and we talk about it, and I'm again making this very, very basic. So you talk about the dominant allele is actually a brown eye and the recessive allele is blue. And most of the time we talk about recessive, it's actually a lack of something. So I have blue eyes. It's because my body, my cells don't actually produce enough of what's called melanin in your eyes to make them darker in color. So blue eyes are actually a lack of pigment, which is why when you're out in the sun, people with blue eyes, they tend to like, oh, you turn the lights too quick, or I want it on the sun, and it's sensitive because your eyes aren't able to reflect as much light from your irises. Brown eyes, you only need one dominant allele versus two. And I'm going to kind of get into that in a minute. You want to do the next slide, Mr. Shooter?
Mm-hmm.
Okay. And so if we go back to that like purple and white flower that they were showing on those chromosomes, okay, purple for flower color, at least in this example, is dominant. White is recessive. So if you look at the letters underneath, and this should be familiar if you had Mr. Beichler, we know that he talked about this a little bit. So we're kind of going in with that idea, is that if you have one of the dominant alleles, we represent that by a dominant or uppercase letter. So it's a capital letter. So if you have just one of those, then you're going to have that color purple. If you have one purple, one big B in this instance, and one little b, you still have one of those big ones, so you're going to be purple. We call that, when you have one dominant and one recessive, we call that heterozygous. So hetero means different. Okay, homo means the same. So if you are heterozygous, you have two different alleles. If you are homozygous, that means you have two of the same alleles. But you can be homozygous dominant, which means you have two dominant alleles, or you can be homozygous recessive, which means that you have two recessive alleles. And you'll notice from the picture, the only way that we will see the recessive trait is if you have two recessive alleles.
And we're talking about heterozygous, not heterozygotes. Right?
I forgot about that one.
Heterozygous, one dominant, one recessive. I do want to make a comment. Earlier in the chat, Eli wrote, "Sucks to know that I was the fastest out of my brothers and sisters." And I'm guessing that comment came up from here. And so I will say this. No, Eli, you were not faster than your brothers or sisters. Your brothers and sisters were different cells. You were, however, faster than the million other sperm cells that you swam against that day. And so everybody that never feels like, if you're out there and you feel like you've never accomplished something in your life, just keep in mind, you were the big winner that day. You all won out over millions of other cells. So you have. All right. Who's this? This is me. Genotype versus phenotype. So here's the last piece of this. And Mrs. Smith talked about it a little bit right here. Because she talked about homozygous dominant, homozygous recessive, and heterozygous. And so what it comes down to is you're seeing two things on there. You're seeing genes, like the big B, the big B, the big B, the little B, the little B, the little B. Those are your genes. Those are what your chromosomes have. That's your genotype. But that's not necessarily what we see. Because if you have brown eyes, I don't know if you carry a cell or a gene for or an allele for blue eyes. Maybe you do. Maybe it's hidden. Maybe you're heterozygous. Right? So we have two different things that we need to talk about. We need to talk about what your genes are. Levi's, whatever. Now, what your genes are, and we need to talk about what your phenotype is. So your genotype and your phenotype. And so genotype is the combination of alleles in an organism inherits from its parents. That's what you got. I got one gene for brown eyes. I got one gene for blue eyes. Your phenotype is what we actually see. And this is really bad, but I always think phenotype for physical appearance, and they have physical here. And I don't know why, but I always think of that song, Let's Get Physical. When I see that, and I say physical, right? Like physical appearance, right? Physical pH, phenotype. That's what we see. Is the flower purple? Is the flower white? Do you have blue eyes? Do you have brown eyes? Do you have blonde hair? Do you have red hair? Are you tall? Are you short? Those are your physical appearance. Your genotype is what genes that you actually carry. And so Yoda would say, my phenotype aren't that great. but I have enough intelligence to make your genotypes look like a joke. I don't know. It's terrible. So we finish up here? Is this still me?
I mean, we can talk about this together, I guess. So, this should look familiar, at least somewhat. Anybody remember? Well, I mean, it's been in the chat already today. But what we're looking at here is a Punnett Square. And so the one thing I'm going to say about a Punnett Square is that when people look at this, they think that this is, this is going to tell me what my offspring are going to be. But I want you to remember that a Punnett Square is a probability tool. It shows you what the probability is of a certain phenotype or genotype. And so if you were to look at this, you see the male pollen up at the top that is purple. And its genotype is big B, little b. Those were the two alleles it got from its parents. So when people say you look like they're your grandparents, it's because your parents' genes are from your grandparents who are from their grandparents, et cetera, et cetera, et cetera. So you got one from mom, one from dad. For the purple flower male, it's got big B, little b. From the female flower, where it has the pistil, okay, again, and going back to plant parts, what was that, 6th grade? From big B, little b as well. So the female also has big B, little b. So the first thing I want to know, because they're both the same, is that I was asking you, what type of genotype is this? What is it? Big B, little b.
So what type of genotype is it? And I'm going to help them just a little bit.
Cheater. Pumpkin eater.
Just reminding them a little bit.
Big B, little b. One of each. Great, Annie. Heterozygous. Okay? So they are both heterozygous. So this is kind of I always think of it as Battleship. So when you're filling out a Punnett Square, you fill it out by taking the pieces and going together. So if you're playing Battleship, you might go, oh, B7, and you're trying to figure that out. So here we're going the two together. So the first left-hand side is big B, which is what? What is the genotype for that then? Somebody just brought up a new math comment, and I can't even reply to that. So.
Yeah. Lag kills me.
I know. I just remember that when I taught 8th grade, I had people that told me that was so much faster, and I proved them wrong every single time. Anyway.
So Radhika and Jessica together have it right.
Great. And Cohen has it right himself. So you have to put both in there. Yes, it's homozygous, but it's homozygous dominant. And the reason that you have to put both is that if you look down in the bottom right-hand corner, that one is 2 little b's, and that would be the homozygous recessive.
Mm-hmm.
And then the other two are heterozygous. But here's the thing. When we talk about genotype and phenotype, you have potentially different genotype percentages than phenotype percentages. For example, if I look at this, 3/4, the phenotype would be purple. But if I talk about genotype, 1/4 is homozygous dominant, and 2/4 or 1/2 is heterozygous. Now, the homozygous recessive and the phenotype for that on white both happen to be 1/4 or 25%. And again, what I want to point out is that these are ratios, they're possibilities. So if this offspring has one white flower offspring, it has that same chance every single time. every single offspring has the same chance. So you could potentially have that 1/4 every single time. Now the chances are lower probability wise, but you could have all white flower offspring. It's not saying, oh, you got one, so the other ones must be the other ones. It's not the way that it works.
Right. So that's kind of it. And so you can guess where we're going to get into next, that we're going to get into talking a little bit about Punnett Squares and figuring out how to do this. Probably not as deeply as we would have if we were still in the building. But that's what we're going to look at this week. So this week, we're looking at least the beginning of this week, we're looking at genetics, and we're trying to figure some of that out. With 15 days left, one of the other topics I know we want to get to is probably evolution. Which I think will be interesting. But anyway, we got some time. Oh, we're done with that. So the moral of the Punnett Square, you see somebody that you find attractive, make some mental Punnett Squares.
Terrible.
All right, ladies and gentlemen, that's it for us. Nothing to do tonight. We'll see you tomorrow at 10:30. Nothing to do if you're done with everything. If you're not done with anything, spend some time on bio. But we'll see you tomorrow at 10:30. As always, love you. Wash your hands.
Bye-bye.
Bye-bye.